Hello viewers, let us discuss the anatomy of testis. At the end of this presentation, the learners will be able to know what is testis. They will be able to know the location and external features of testis. What are the coverings of testis? What is the structure of testis? Histology of testis. They will also be able to know the blood and nerve supply of testis and will be able to describe the applied anatomy of testis. The testis is a male gonad. It is homologous with the ovary of the female. It's a paired organ. It has got right and left testis. The testis is suspended in the scrotum by the spermatic cord. Left testis is slightly lower than the right, and the testis is placed obliquely with the upper pole tilted forwards and medially. The shape and size of the testis. The shape of the testis is oval. It is compressed from side to side with 3.75 centimeters long. Vertically, it is about 3.75 centimeters long. The anterior-posterior diameter is about 2.5 centimeters. That is 2.5 centimeters broad from before backwards. And its thickness from side to side is about 1.8 centimeters. Its weight is about 10 to 15 grams. The external features of testis are: it has got two poles, upper pole and lower pole, two borders, anterior border and posterior border, two surfaces, medial and lateral surfaces. Two poles, two borders and two surfaces. The poles are upper and lower pole. The borders are anterior and posterior, and the surfaces are medial and lateral. The details of external features are like this. The poles are smooth and convex. The upper pole is attached to the spermatic cord, and the appendix of testis is a small oval body that is present over the upper pole. The anterior border is convex and smooth and is covered by the tunica vaginalis. It's a covering of the testis. It is covered by tunica vaginalis. The anterior border is convex and smooth and is covered by tunica vaginalis. The posterior border is partially covered by tunica vaginalis, and it is straight as compared to anterior border. The anterior border is convex, but the posterior border is more or less straight and is occupied by the epididymis. The lateral part of the epididymis is separated from the testis by a cavity of the tunica vaginalis, which is referred to as sinus of the epididymis. Now this is a reflection spot. Answer this question by pausing the video. Once you are ready with your answer, you can resume the video. The question is, where is the testis located and what are the external features of testis? You must have answered the questions. The testis is suspended in the scrotum by the spermatic cord, with left testis slightly lower than the right, and is placed obliquely with the upper pole tilted forwards and medially. You must have also answered the external features of testis, that it has got two poles, upper pole and lower pole, two borders, anterior and posterior border, and two surfaces, medial and lateral surfaces. Now let us see what are the coverings of testis. The testis is covered by three coats from without to inward, from outside to inside. There are three coats. They are Tunica Vaginalis, Tunica Albuginea and Tunica Vasculosa. The Tunica Vaginalis is formed by the processus vaginalis, which is a peritoneal tunnel that passes through the inguinal canal into the scrotum, and at the lower end of which the testis develops. The processus vaginalis is invaginated by the testis from behind. The testis invaginates the processus vaginalis from behind like this. The testis slowly enters into processus vaginalis part by part. From first, the anterior border will enter into processus vaginalis. So the anterior border is completely covered by processus vaginalis, forming the tunica vaginalis. The posterior border, is devoid of tunica vaginalis or is partially covered by tunica vaginalis. Now you can see this. The processus vaginalis is completely invaginated by the testis, which results in formation of the tunica vaginalis with two layers: parietal layer of processus vaginalis and visceral layer of processus vaginalis. These are the two layers of that tunica vaginalis. The posterior border, you can see, is devoid of the tunica vaginalis. Only the anterior border is covered by tunica vaginalis with a parietal and a visceral layer. The tunica albuginea is white fibrous coat which is dense and is covered by tunica vaginalis from outside. The posterior border is thickened to form what is called as mediastinum testis, and septa from mediastinum testis enter into the substance of the testis and divide the testis into 200 to 300 lobules. This is how the tunica albuginea divides the testis into 200 to 300 lobules. Now you can see the tunica albuginea is forming septa which divide this testis into 200 to 300 lobules. And each lobule is lined by the vasculosa coat, or the innermost coat, which is vascular coat, is called as tunica vasculosa. The structure of the testis. Now, the structure of testis mainly consists of seminiferous tubules, which are about 60 centimeters long and 0.2 millimeters in diameter. They are more than half a meter in length, but they are highly coiled. Due to the highly coiled structure, they are able to be placed within the testis. They are lined by the cells of spermatozoa in different stages, and 2 to 3 seminiferous tubules in each of the lobule join to form 20 to 30 straight tubules. These straight tubules enter the mediastinum testis, which is part of tunica albuginea, and forms a network of tubules which is called as rete testis. From rete testis, about 12 to 30 efferent ductules arise, and these efferent ductules near the upper pole of the testis enter into epididymis. The efferent ductules become highly coiled to form a lobule of head of the epididymis. Efferent ductules end in a single ductule, single duct, which is highly coiled to form the body and tail of the testis. The seminiferous tubules join together to form the straight tubules. The straight tubules form a network of tubules which is called rete testis in the mediastinum testis. From mediastinum testis, efferent ductules arise and they enter into the head of the epididymis, and from this head of the epididymis, these 12 to 30 efferent ductules join to form a single duct which is highly coiled, and it forms the body and tail of the epididymis. Here is the reflection spot. Pause the video and answer these questions. What are the coverings of testis? And name the parts of structure of testis. You must have answered the questions. The coverings of testis are Tunica vaginalis, Tunica albuginea, and Tunica vasculosa. The parts of testis, you must have answered that it consists of seminiferous tubules which join to form straight tubules. Straight tubules will form network of tubules which is called retrotestis. From retrotestis, the efferent ductules arise which go into head of the epididymis. From head of the epididymis, 12 to 30 tubules join together to form one long tube which is very much coiled, and that forms the body and tail of the epididymis. So the structural sequence of the testis are seminiferous tubules, straight tubules, retrotestis, efferent ductules, and epididymis, head, body and tail of epididymis. Now let us see the histology of testis. The histology of testis consists of seminiferous tubules arranged in 4 to 8 layers. The cells are of two types: spermatogenic cells and supporting cells, which are also called as sustentacular cells or Sertoli cells. The spermatogenic cells include spermatogonia, primary spermatocytes, secondary spermatocytes, spermatids, and spermatozoa. The cells of Sertoli are tall and columnar cells extending from the basal lamina to the central lumen. There are interstitial cells of Leydig which are found in small clusters in between the seminiferous tubules, and they secrete testosterone. Arterial supply of testis. The testicular artery is a branch of abdominal aorta arising at the level of L2 vertebra, descend on the posterior abdominal wall and reach the deep inguinal ring and enter the spermatic cord. Through spermatic cord, they enter into the testis. The venous drainage of testis. Veins emerge from the testis and they form pampaniform plexus. The plexus are arranged in 3 groups. The plexus condense to form 4 veins at the superficial inguinal ring, and they condense to form again 2 veins at the deep inguinal ring and accompany the testicular artery. Finally, 1 vein on the right joins inferior vena cava and 1 vein on the left joins the left renal vein. So initially there are venous plexus which result in formation of 4 veins. After passing through the inguinal ring, they get converged into 2 veins, and finally there is only 1 vein at the deep inguinal ring that accompanies the testicular artery. On the right will join inferior vena cava and on the left join the left renal vein. We will discuss further in our next